Hi everyone, I am Somnath Karan. Today I will present you Response Adaptive Randomization or RAR. Before I state this presentation, I will show you what are the challenges in our traditional randomization. In our traditional randomization, we do the equal randomization. Like if you have three treatment, so we randomize like 1:1:1 or 2:1:1 or 5:2:1 like that. Think about if you have 10 more than 10 arm or multiple arm or multiple treatment, and our objective is the find out the effective treatment as soon as possible. And this thing is happening in oncology or rare disease trial. And suppose we have 10 arm or treatment, and we doing the our traditional randomization, and we randomize in equal way. So the we assign the same number of patient in our 10 arm and after the study end we analyze the all data and find out the which treatment are the effective treatment. So in this procedure, will see the more we see the most this more 2 to 5 treatment will be perform better, or other are not performing well. So we enroll more number of patients in ineffective treatment, that we can control using the interim analysis, right? But this thing is not doing our traditional randomization. So it will be the ethical concern that we enroll ineffective treatment more patients, and it is require also large sample size, and that not possible in our oncology and the rare disease trials for few diseases. And it is inefficiency also because more number of patient is enrolled in the ineffective treatment, and less number of patient has enrolled in effective treatment. So let's start the presentation. What is ARIA? The response adaptive randomization is a type of randomization where the probability of assigning new patient to into the treatment is based on the response or outcome of the patient that already enrolled in the study. Suppose imagine if you have a treatment A and B. First, we have equally randomized, that is 50% patient to treatment A and 50% patient to treatment B. And after the interim analysis, we saw the A is better efficacy than B. Then we can adjust the randomization probability A to more and B to less. Like here, I adjust the 70% to A and 30% to B to allocate the new patients. What is the common method of REL? Common methods are randomized play the winner zone, covariate adjusted response adaptive design, doubly adaptive biased coin design, and the Bayesian response adaptive randomization or BRAR or Bayesian framework of RER. I only focus on this Bayesian response adaptive randomization to this presentation. And or what is the Bayesian area? This is the response adaptive randomization. The randomization probability is updated based upon the Bayesian framework. Let's see how the process BRR process going on. Initially we defined all our study into objective, endpoint, and what are the treatment, what are the study period, etc. After we define our primary endpoint, and we will consider the power distribution of our primary endpoint, and start enrolling the patients and randomize equally first into the treatment group, and collect the interim analysis to update the posterior distribution of our primary endpoint using the Bayesian hierarchical Puppet model, and compute the allocation probability to assign the new patients into the study and monitor whole progress and check the futility and successful criteria. If it is satisfied, we consider end the trial, and otherwise we continue our trial until our required sample size is not achieved. This is the spaghetti plot of BRAR. This plot showing how the randomization probability distributed over the time. Here, this plot is generated using the simulation study. This is the 10 arm I have taken. Here, the x-axis represent the time and y-axis represent the randomization probability. Here 10 arm. So the initially we do the equal randomization. So the randomization probability of each arm will be the point 0.1. After the first interim analysis, we see the randomization probability is changes, and we see how the it is changes over the time. After the few interim analysis, we see that the three arm has a more epic high probability and other are very low probability. So we continue with these three and we can drop this other arm from the study. This Bayesian areas is already applied in the few study like battle study, I spell to spin control and NTV trial also. This battle study is the biomarker integrated approach of targeted therapy of lung cancer elimination. I will show you how the Bayesian area framework is applied in this study. This battle study is the phase 2 umbrella trial and patient has with advanced non-small cell lung cancer or NSCLC patient has enrolled and had the four treatment eldotinib, bendatinib, sorafenib or combo 1 eldotinib plus bexarotene is tested over the 5 biomarker group, marker group 1 to marker group 5. Here, the main objective of this study was the give personalized medicine using the Bayesian RAR method. The primary endpoint was 8-week disease control rate. Disease control rate is DCR. So disease control rate is the ratio of number of patient disease control divided by total number of patient has enrolled in evaluated. The futility criteria of the study was probability of A to DCR exceeding the 50% of given data will be less than 0.1, and the successful criteria was probability of A to DCR exceeding the 30% of given data will be greater than 0.8, and the type 1 error was 20%. Here the high type 1 error was taken because of high power and more and high probability of a find the effective treatment and low probability of overlooking ones. In this battle study, the total 341 patient has enrolled and 255 patient has randomized, 97 using the equal randomized and 158 patient using the Bayesian RAR method. And this graph showing the how the randomization probability is distributed over the marker group by treatment group. For the marker group 1, the compo 1 is high randomization probability as compared to the other three treatment. So we can say that the combo 1 is more effective treatment compared to other treatment in for marker group 1. For marker group 2, Bendatinib is very low randomization probability, and other hand the sorafenib is very high randomization probability. So you can say that Bendatinib is very less effective treatment for marker group 2 and sorafenib is high effective treatment for marker group 2. Similarly, for marker group 3, Betatinib and combo 1 is a very less ineffective treatment. Other hand, the sorafenib and the eldotinib is very high effective treatment. Similarly, marker group 5 bendatinib again is ineffective treatment, and other hand the sorafenib is high effective as compared to the other two treatment. Here the marker group 4 is not showing because of very less number of patient has enrolled in that particular marker group. This table showing how the 8-week DCR status by treatment and marker group. Here the numerator number showing the number of patient with disease control, and denominator number showing the number of patient has evaluated, and bracket one showing the 8-week disease control rate. And the highlighted cell showing the effective treatment means the probability of 8-week DCR exceeding 30% of giving data will be greater than 0.8. For marker group 1, the bendatinib and the combo 1 is effective treatment. For marker group 2, sorafenib is effective treatment. And marker group 3, the eldotinib and sorafenib is effective treatment. For marker group 4, again the combo 1 is effective treatment. For marker group 5, the combo and the sorafenib is effective treatment. And if you see the marginal DCR of the treatment is for sorafenib is 58% and combo 1 is 50% and bendatinib is 33% and the eldotinib is 34%. So the sorafenib is more effective treatment over the marker group as compared to the other treatment. So using this BRAR method, this battle study achieved 46% DCR over the 224 patient using the Bayesian Response Adaptive Randomization method. And this BRAR method provides the personalized medicine as for marker group 2, the sorafenib is high effective treatment, other are ineffective treatment. So we enroll more number of patients and that particular marker group. So it is also the inefficiency as we enroll more number of patients and to the effective treatment and less number of patients in ineffective treatment. So what is the advantage of BRAR? Ethical advantage: prioritize effective treatments, reduce the exposure to less effective treatments once, and statistical advantages: improve the statistical power, smaller sample size required as compared to the our traditional randomization, reduce the variances also. And this Bayesian RAR method is useful in oncology and rare disease trial where we need the personalized medicine. Challenges and drawback of BRAR. Challenges require real-time data analysis as we updating the Bayesian model time to time. And risks of type 1 error inflation also, complexity in design and execution, choosing the biodistribution should be the tricky. And drawback: estimation bias may be happen, consideration long-term endpoint will be challenging as our main objective is to give the personalized medicine, so we need to update the posterior distribution and and compute the allocation probability um, to very short time. If you take it is take our endpoint is take long time, then in this model is updated thing very taking very long time. So our main objective is not more effective way work in that case, and this model also not considered the toxicity of the drug. Key takeaways and future direction. BRR improve the patient care and safety also, provide the strong result with efficient trial. This method also support the progresses in cancer and rare disease trial and open opportunity for personalized medicine. Thank you for listening. If you have any question, you can comment on this comment here. Thanks you for the for listening this presentation. Bye.